Hi, I'm Koji from SCAN, and Aseptic Technology is a member of SCAN. And today I would like to introduce the from R&D to commercial production key decision in GMP manufacturing. So let me introduce who we are, the SCAN and Aseptic Technology. The SCAN is a Swiss company established in 1968, and we're the global market and technological leader for the isolators for aseptic production in pharmaceutical and biopharmaceutical area. And cell and gene therapy, we do have the different product portfolio, as you see in here, which is, of course, as Aseptic Technology, we have closed vials together with their filling system and the surrounding devices. And we do have the process solution isolator, which will be applied to the upstream, downstream of whole cell and gene therapy, including the fill finish. And we do have the good practice design consulting services and aseptic manufacturing service in Switzerland now. And here's our global customers in pharmaceutical field. And today I like to talk about the product life and what's going to be the biggest milestone when you think about your life of the product. So when you see the market situation of the ATMP, as you see in here, now the 75% of the product under the development is in the R&D to preclinical. So this is the market situation right now, and then some of us already attended the morning presentation already, and then their pipeline in the companies they presented this morning is really like this field R&D to preclinical. And then when you talk about the life of product, you have to think about the end-to-end platform, which is starting from R&D, process, product, direct, container, and facility, capability, team, process machines, quality management system, regulatory affair, automation, life cycle maintenance. And then finally, at the end of the clinical stage 3, you can launch your product, the product to the market, which is a commercial adoption you have to think about. And the first milestone we like to focus on is the product direct contact, the parts and the filling. So the condition of the product direct contact parts is the particle-free within the defined level, and toxin-free, and sterilized, and stable to product, and stable for storage. And here is a simple comparison of the different type of the product, the container. So open vial was a plastic COC, COP, and the closed vial from Aseptic Technology and the bags. And what we have to focus on is the particle level and the toxin level. So when you see generally the open vials, the particle is controlled under the grade C, the manufacturing, the clean level. And the bag is the same, manufacturing the grade C. But Aseptic Technology closed vial is the only the plastic vials which is manufacturing the grade A. Plus, once it's assembled together with the TPE stoppering, it's already sterile inside. So at the time of the shipment of this closed vial, at the time of the use, it will be kept sterile all the way. And the next point is the endotoxin-free. So the open vials, generally, endotoxin-free is not guaranteed, but usually it's measured before the shipment to make sure that the endotoxin is 0. And then the AT closed vial is, again, the only the vials which we can guarantee the endotoxin-free. And then back is also the same as the open vials. So this is the typical open vials. It's the West Pharmaceutical Dichocrystal CZ vials. And this is the AT closed vials design. As you see in here, a different point of the between open BIOS and the AT closed BIOS is this TPE stoppers. So when it's manufactured and assembled with the stoppers, so this is the biggest difference between the open BIOS and AT closed BIOS. And also, Aseptic Technology has the filling technology, starting with a manual filling to the robot filling, up to the high-speed robot filling. And this is the ideal solution for this industry, because when you have to select the scalability, scale out or scale up, so the filling technology has been using the same filling system and same closing system, so you can easily select and decide the scale out to scale up the approach with a different type of the filling system. And as I said, the Aseptic Technology also has the surrounding devices, which is Celix and the AT port. And the Celix is the integrated equipment to mix the fill finish product itself and cool, to keep it cool. So the quality of the product itself will be maintained in ideal the condition right before the fill finish. And then the product will be directly connected to the either ice bed or the biosafety cabinet through the AT port. So this is the AT port is the sterile connection in aseptic manner. And the next milestone is the investment. When you go to the R&D application and then the start the clinical study, and then you have to think about the investment, because the lab scale setup will be shifted to the production setup. And when we see the market situation now, these are the number of approved products in the global market. And the approval ratio under the total development, the product is 10% to 20%, which is really high number compared to the traditional injections. But also, on the other hand, 80 to 9% fail. And this is really surprising, the information maybe. The 60% of this 80% to 90% fail, it's not because of product efficacy or the safety. The 60% fail is because of the scalability, cost, manufacturing capability, GMP compliance, and market adoption. So this is simply technical, the point which we could solve if we prepare well at the R&D to clinical the study. So how we can avoid this technical failure to bring your product to the successful commercial launch? There are three different the approach at this point. One is the merged by big company, and the second one is a self-funding, and the third one is a partnership to CDMO. And the merged is easy. You will sell the company, and then the owner will be like rich people, so happy ever after. So this is just the information. And the potential solution number two is self-funding. And just make it simple, the self-funding, you have to think about the approx 50 to 100 million euro investment. And then this investment will not be returned for 5 to 8 years until you launch the product. So it's money-eating solution. And the third solution is a partnership to CDMO. And this is really ideal because work with a professional team to define this, the scalability approach and efficient manufacturing cost. And then we can realize the best approach for the scalability and the cost, and you can achieve the massive cost reduction. And you have to think about the technology transfer, load to GMP. And the milestone number three is the clinical production, commercial production compliant to GMP. When you talk about the GMP mindset, the GMP is not the one way. GMP has to be circle around starting from R&D to process development, and then GMP, the production, and coming back to the R&D. So I can give you the good example. If you select one of the vials at the R&D phase, which you realize at the commercial stage you like to use a different vials, but it's too late. You cannot to bring everything back to the clinical stage 1. So you really have to be careful and also be ready to select the right solution for your product from the beginning to commercial production. So therefore, together with the CDMO capability, what you have to think about is a good practice design consulting, production scaling, and also the next generation manufacturing. Then the next one is a process machine fully compliant to cGMP. And when you think about the different the cell and gene therapy processes and an aseptic open process, when you focus on the aseptic open processes, so these blue part is the aseptic open process. And when you see the warning letter from authority for the traditional injection production, and there are the 3 different ones. The transfer of direct impact container between A and B is a cross-contamination and a microbial contamination risk. And manual open process to direct impact to sterility is huge risk of microbial contamination, and it is not allowed. And impact on unidirectional airflow is cross-contamination and microbial contamination risk. So these 3 points, when we compare the biosafety cabinet, which is typical lab scale setup, together with the ice splitter, which is the typical production for the aseptic, the injections. When you see the open process with the biosafety cabinet, what you have to do is the open process will be done in the biosafety cabinet, but to use the process machine such as a centrifuge incubator, you have to bring out your the product from grade A to the grade B, and after process machine, you have to bring it back to grade A. So this is the really risky, risky to point out the transfer of direct impact container between A and B. And instead, the ice splitter, you can integrate the whole process machine into the ice splitter. You don't have to bring out or bring all the materials back and forth. You don't have to do it. And then here's another one, the direct access. Biosafety cabinet, you need to have a direct access to your the process. Instead, the ice splitter is covered with the stainless steel and glass and gloves. You will not allow to have the direct access. All access is anyway all the time through the gloves. And here is the unidirectional airflow. So when you see the unidirectional airflow with a biosafety cabinet without the operator, it's the ideal like this. But when you have the operator to start the operation, it will have creating the turbulence. Instead, the ice splitter is fully closed. Therefore, even operators start working inside the ice splitter, the unidirectional airflow will not be disturbed. This is some ice splitter configuration for the upstream, downstream of the mesenchymal stem cell or the stem cell application, or either the aseptic open process of the CAR-T. And also we do have the fill finish ice splitter configuration. And this is one of the case study with Takara Bio in Japan. And they're using the ice splitter for the open process of the CAR-T process, which is the medium preparation and the recovery formulation. And this is the comparison data between the biosafety cabinet production and ice splitter production. And as you see in here, the expansion rate and survival rate, immunophenotype, and transduction rate, and the probios copy numbers, those are the really comparable the quality. So ice splitter that you can use perfectly with the CAR-T process. And the biggest point is environmental monitoring. with the ice well, there is no access of the operator directly to the process, which means the risk of the microbial contamination, cross-contamination is significantly low. And this is the IPS in ice well. So this is also the same, the quality compared to the biosafety cabinet, which is already proven. So as in here, starting with the R&D to commercial, the aseptic technology and the scan can support your the whole road to bring your product to the commercial production. Thank you.